ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన ఎన్ఎస్ఆర్ ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి, జేఈఈ అయినా, నీట్ అయినా తిరుగులేని విద్యా సంస్థ ఎన్ఎస్ఆర్ ఇంపల్స్.
Hello students, now let us continue our discussion with respiration. Where are we in respiration? So for the last class we discussed about types of respiration. We discussed thus aerobic respiration and anaerobic respiration. In anaerobic respiration the first step is glycolysis. Aerobic respiration also the first step is glycolysis. In anaerobic respiration we discussed about glycolysis and fermentation. Now by the end of glycolysis pyruvic acid is produced. Fermentation, if you take, it is again of two types we discussed for the last class. What are they? One is alcohol fermentation. If it does not happen then it may be lactate fermentation. Fermentation process we find in case of lower organisms like microorganisms like yeast and bacteria and other organisms. In alcohol fermentation what is produced? Acetaldehyde and the end product is ethyl alcohol produced from pyruvic acid. In lactate fermentation the end product is lactic acid produced from pyruvic acid. So in human skeletal system during exercise, when humans are doing exercise, deep tissues may not get sufficient amount of oxygen, then they undergo anaerobic respiration and produces lactate or lactic acid. So that is what we discussed under anaerobic respiration. Then for aerobic, anaerobic, the common biological, universal biological process is glycolysis. So we discussed glycolysis for the last class and that is very important. Glycolysis itself is also called EMP pathway, also called as a cellular respiration pathway, a biological pathway, core respiration pathway. That is first step what we completed. It's nothing but glycolysis. This is the first step. So in glycolysis what happened? So we studied the glucose, one molecule of glucose, nearly 10 steps we studied and produced with 2 molecules of pyruvic acid. So each one having 3 carbons. So glycolysis is a common pathway for aerobic respiration and anaerobic respiration. And first step glycolysis we completed. Now we are in the discussion about aerobic respiration. Then after glycolysis let us continue the second step called as link reaction. Now we have to continue with link reaction, followed by what is the third step we have to continue? Krebs cycle. Then followed by the fourth step is nothing but ETS, electron transport steps, electron transport system. So aerobic respiration altogether it has 4 stages or 4 phases we call. First one is glycolysis, that's what we completed. Second one is link reaction. Third one is Krebs cycle and the fourth one is electron transport system. So we completed now the glycolysis. End product produced in glycolysis is nothing but pyruvic acid. Now let us continue with what is called second one called link reaction. So let us take second one is link reaction. Now this link reaction also called as oxidation 2 and decarboxylation 1 . So this is also called as, why it is called oxidation 2 and decarboxylation 1? In glycolysis, did you remember there is a step where G3P produced into 1,3-biphosphoglyceric acid. Did you remember? So here I will just tell you glucose 6-phosphate, fructose 6-phosphate, fructose 1,6 biphosphate, and from here DHAP and here G3P G3P gets converted into 1,3-biphosphoglyraldehyde. Did you remember? So what happens here? There is a production of NAD plus 2 molecules are produced here, 2-NADPH2. So what is this step? This is called oxidation number 1. So oxidation number 1. Oxidation number 1 produced with 2 molecules of NADPH2. This happened at the time of glycolysis. So that is very important for all of you to remember. Oxidation number 1 takes place where? In the glycolysis. Where it takes place? Conversion from G3P to 1,3-biphosphoglyraldehyde. That is the first oxidation step in aerobic respiration. This is oxidation number 1. Then glycolysis we completed, right? Then link reaction, here this is referred as second oxidation step, and here carbon dioxide is also released. So this is called decarboxylation number 1. So in the total aerobic respiration there are 6 oxidation steps and there are 3 decarboxylation steps. So we have to take the count for all 6 oxidation steps. We have to see all 6 are not at one place. In glycolysis oxidation 1 happens, oxidation 2 happens in link reaction, oxidation 3, 4, 5 happens, 6 happens in Krebs cycle. So you need to be very clear and cautious in listening the respiration. So this is first oxidation in glycolysis. I hope this you understood. So this itself is also called oxidation 2, and here there is first step called as decarboxylation occurs. So this is also called as oxidative decarboxylation step. Itself is also called oxidative decarboxylation, but try to remember this is oxidation number 2, and here itself decarboxylation begins, and this is first step of decarboxylation. We will see now, right? And itself is also called as gateway path, gateway step. Why it is called as gateway step? So here just now we studied glycolysis. Glycolysis occurs where? In the cytoplasm. Glycolysis occurs in cytoplasm. Very important. May have sometimes you will get it in the form of a question. Where does glycolysis takes place? Glycolysis occurs in cytoplasm. Then followed by what is their next? Link reaction. So link reaction, why do you call this link reaction? Because from cytoplasm what is produced here? Pyruvic acid. PA I am writing. What is produced here? Pyruvic acid. By the end of glycolysis pyruvic acid produced. This enters into mitochondrial matrix. It enters into mitochondrial matrix. Very important. What enters? Pyruvic acid produced at the time of glycolysis, where in cytoplasm now enters into mitochondrial matrix. So in mitochondrial matrix, here pyruvic acid enters into mitochondrial matrix and undergoes link reaction, and then inner mitochondrial membrane what occurs is called Krebs cycle. Krebs cycle, it's really a big cycle. In this so many intermediates are there. So for glycolysis here it occurs in all, it also occurs in the same mitochondrial, inner mitochondrial membrane. So, for glycolysis occurs in the cytoplasm, Krebs cycle occurs in the mitochondria. For these two, the connecting step, a small connecting step, it's called link reaction. Okay, glycolysis occurs in cytoplasm. Krebs cycle occurs in mitochondrial membrane. Now, in between there is one small step which makes connection of glycolysis and Krebs cycle. That small step itself is called link reaction. It is a very small reaction where it occurs mitochondrial matrix. So, remember, link reaction occurs in mitochondrial matrix, very important. So, with this link reaction, it enters into mitochondrial matrix. That is why it is also called as gateway step. Here oxidation 2 takes place, decarboxylation 1 takes place, very important, and it is a connecting link between glycolysis and Krebs cycle, because glycolysis occurs in cytoplasm, Krebs cycle occurs in the mitochondria. These two, for this Krebs cycle, gate opens through link reaction. That is why itself is also called as gateway step, right. So, remember, glycolysis occurs in cytoplasm, link reaction occurs in mitochondrial matrix, link reaction connects with Krebs cycle also occurs in mitochondrial matrix, right. Now, let us continue with the link reaction. Now, if we take the link reaction here, what happens? What is the end product of glycolysis here? Pyruvic acid. So, here let us take 2 molecules of pyruvic acid. 3 carbons, right. What is the end product of glycolysis? 2 molecules of pyruvic acid shifted from where to where? Cytoplasm, entered into mitochondrial matrix. Now, it is undergoing link reaction. Let us see now. So, if you look at in this case, so here it is like this. Now, and it produced with here the 2 molecules of NAD plus get reduced to 2 molecules of NADPH2. So, here 2 molecules of NADPH2 is produced, and here 2 CoA molecules are added, and there is a release of 2 carbon dioxide. Okay. Here 2 CoA, it is nothing but what you call factor, prosthetic group. In enzymes we studied cofactor, prosthetic groups are added, 2 CoA is added, and that produces and releases 2 molecules of carbon dioxide. That means 2 NADPH2 is produced and 2 carbon dioxides are released, 2 molecules of carbon dioxide is released. This is the first step where carbon dioxide is released out. That is why it is called decarboxylation number 1. Here NAD plus get reduced to form 2 NADPH2. This is oxidation number 2, right. And as a result of this, it produced with the compound called as 2-acetyl-CoA. 2-acetyl-CoA. This is having 2 carbons. Why it is having 2 carbons here? In this, from each pyruvic acid, 1 carbon is lost in the form of carbon dioxide. 2 molecules are there, so 2 carbons are lost in the form of 2 carbon dioxide. So, here 3 carbon pyruvic acid converts into 2 carbon acetyl-CoA. For each pyruvic acid, 1 carbon is lost in the form of carbon dioxide. So, out of 3 carbons, 1 carbon is lost, so 2 carbons are produced. As 2 molecules are there, so 2 carbon dioxides are lost and 2 molecules of acetyl-CoA with 2 carbons is produced. Now, this happens in the presence of a multi-enzyme complex. Multi-enzyme complex. This happens with the help of a multi-enzyme complex. What are that multi-enzyme complex? There are so many factors, like magnesium is one factor, right. Apart from this, even if I am writing here TPP, thiamine pyrophosphate. TPP. Then apart from this, lipoic acid, CoA, FAD and NAD. So how many cofactors? Multi-enzyme complex, it includes altogether 6 cofactors. It includes 6 cofactors. And what is that multi-enzyme complex? Here we have the multi-enzyme complex, what we find here, it's nothing but pyruvic dehydrogenase. What is that? Pyruvic dehydrogenase. This is a multi-enzyme complex. What is the name of the enzyme? Pyruvic dehydrogenase. Okay, so I'll just repeat here. 2 molecules of pyruvic acid having 3 carbons converts into 2 molecules of acetyl CoA having 2 carbons. Now, while converting here, 2 molecules of CoA cofactor is added and 2 NAD plus is added, that get reduced to form 2 NADPH2. So, here there is a production of 2 molecules of NADPH2. NAD plus get reduced to form NADPH2. That is why we call this step as oxidation 2. And here there is a release of 2 molecules of carbon dioxide. At lastly, even we have to make the count for carbon dioxide. So, at that time we write 2 molecules of carbon dioxide released out in link reaction. That is why link reaction is also called as decarboxylation number 1. Oxidation 2 and decarboxylation 1. That is why this pathway is also called as oxidative decarboxylation. And then here this is also called gateway step, because acetyl CoA now it enters into mitochondrial matrix. Acetyl CoA now it enters into mitochondrial matrix. That is why itself called as a gateway step to enter into mitochondria. Now, so if you look at in this, the 2 molecules of pyruvic acid converts into 2 molecules of acetyl CoA with the help of a multi-enzyme complex called pyruvic dehydrogenase. So, pyruvic acid converts into acetyl CoA with the help of enzyme. What is the name of the enzyme? Pyruvic dehydrogenase. Now, why do you think it's called a multi-enzyme complex? Because pyruvic dehydrogenase, it's not simply one single enzyme, it's a coordination of 3 enzymes. Okay, that's why it's called a multi-enzyme complex. Why? Because pyruvic dehydrogenase, it's a coordination of 3 enzymes plus 6 cofactors. So, first of all, now let us talk about multi-enzyme complex pyruvic dehydrogenase. As I told you, it's a coordination of, why do you call this is a multi-enzyme complex? Because it is coordination of how many enzymes? It contains 3 enzymes. It is coordination of 3 enzymes plus 6 cofactors. That's why it's called a multi-enzyme complex. Okay, I repeat. Why pyruvic dehydrogenase enzyme it's called multi-enzyme complex? Because this complex includes 3 enzymes and 6 cofactors. 3 enzymes works here efficiently in conversion, in converting pyruvic acid into, in transferring into acetyl CoA, and for that it needs the help of 6 cofactors with the functioning of 3 very important enzymes. Now let us start writing what are the 3 enzymes. Pyruvic dehydrogenase includes the 3 enzymes. What are they? Let us start writing one by one. The first one is pyruvic decarboxylase. Let's write small d. What is the first enzyme? pyruvic decarboxylase, and the next enzyme is dilipo or dihydrolipo, anything you can write. Dihydrolipoyl transacetylase. Dihydrolipoyl transacetylase, this is second enzyme, and the third enzyme is also dihydrolipoyl dehydrogenase. Okay, so if you look at pyruvic decarboxylase, first enzyme is pyruvic decarboxylase. Why? Because carbon dioxide is removed. As carbon dioxide is removed here, the first enzyme what plays an important role is pyruvic decarboxylase. Next is dihydrolipoyl transacetylase, lipoic acid. Lipoic acid is plays an important role in transacetylase enzyme in transferring pyruvic acid to acetyl-CoA. Then the third enzyme dihydrolipoyl dehydrogenase enzyme, wherever you find NAD plus get reduced to form NADPH2, here comes into the role called as dehydrogenase. So dehydrogenase will come here in conversion of NAD plus into NADPH2, decarboxylase will come here in removing carbon dioxide, transacetylase converts pyruvic acid, transfers into acetyl-CoA. So multi-enzyme complex, it needs the effective function of 3 enzymes. What are the 3 enzymes? Pyruvic decarboxylase, dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase, all 3 together called as pyruvic dehydrogenase complex. Pyruvic dehydrogenase complex itself is called multi-enzyme complex. At the same time, for the effective function of these 3 enzymes, this enzymatic complex requires 6 cofactors. What are the 6 cofactors here? So let us take one by one. There are 6 cofactors. Try to remember what are the 6 cofactors. Number 1, TPP. What is TPP? Thiamine pyrophosphate. Thiamine pyrophosphate. What is the first one? TPP. TPP, it's nothing but thiamine pyrophosphate, TPP. And second one is lipoic acid. Now this is CoA, is the third one. Third one is CoA. This is called as coenzyme A. This is called coenzyme A, right? And the fourth cofactor is FAD. FAD, it's nothing but flavin, adenine, dinucleotide. FAD, it's nothing but cofactor. Similarly, fifth one is NAD. What is NAD? Nicotinamide, adenine, dinucleotide. Right? So if you can get it, what is TPP? TPP thiamine. Thiamine is an amino acid. Pyrophosphate, you remember in plant growth regulators, auxins, gibberellin, cytokinins, you remember TPP, pyrophosphate we studied there. For which plant growth regulator TPP act as the cofactor or the enzyme or the precursor for the formation of which plant growth regulator? Did you remember? Try to recollect it because one chapter is interrelated with the other chapter, right. So, here this multi-enzyme complex requires 3 enzymes. These 3 enzymes they function efficiently with the help of 6 cofactors. What are they? First one is TPP, thymine pyrophosphate. Second one is lipoic acid. Third one is coenzyme A. Then fourth cofactor is FAD. We all know FAD. What is FAD? F is nothing but flavin, A is nothing but adenine, D is nothing but what? Dinucleotide. You already know in biomolecules, you remember we studied nucleotides, nucleoside. Did you remember? Nitrogen base, sugar. It's called nucleoside. What is nucleotide? Nitrogen base. Did you remember this all? Everything is interrelated. Guys, this concept you can correlate with biomolecules. So, first year. So, this is the first year chapter. What you need to do? Revision. Questions will come interlinked with the first year biomolecules. So, what is that? Nitrogen base plus sugar plus phosphorus. What do you call that? Nucleotide, dinucleotide. And what is this adenine? You know, DNA, RNA, nucleic acids. They are made up of nitrogen bases. What nitrogen bases? Adenine. Which nitrogen? How many nitrogen bases we have? Nitrogen bases, if you remember, we have 2, purines and pyrimidines. Purines are what? Adenine and guanine. What are pyrimidines? Cytosine, thymine and uracil. Did you remember? You need to recollect back the basics of first year biomolecules to continue understanding about respiration. So, here adenine derivative is this. So, which is the adenine derivative of plant growth hormone? Try to recollect it. That means you are connecting plant growth regulator chapter with the respiration with the first year biomolecules. Right, adenine is a nitrogen base. Dinucleotide, you know nucleotide meaning, nitrogen base, sugar, phosphorus. Flavin is nothing but the cofactor here. So, when flavin is a component combined with adenine is a purine, dinucleotide, it is nothing but the component of nucleic acid. When all 3 combines, it forms a cofactor called FAD. Similarly, NAD, N4, nicotinamide, A4, adenine, same thing here. Adenine derivative and dinucleotide combines with nicotinamide act as cofactor. How many became 1, 2, 3, 4, 5? Where is the 6th one? 6 cofactors we have written, we said, right? So, 6th one is nothing but magnesium. Magnesium also act as a cofactor here in multi-enzyme complex to make the efficient functioning of all 3 enzymes. All 3 enzymes, they work with the help of 6 cofactors, together forms a multi-enzyme complex that is called pyruvic dehydrogenase complex, which converts pyruvic acid into acetyl-CoA. So, what are the end products of this? 2 molecules of carbon dioxide is released. 2 molecules of NADPH2 is released and end product as 2 molecules of acetyl-CoA. Right. Now, let us write the reaction, the same link reaction, let us put it in the form of a reaction. So, how to write this? So, let us say that 2 molecules of pyruvic acid, each having 3 carbons, plus. So, 2 molecules of CoA is added, 2 molecules of CoA plus 2 molecules of NAD plus get reduced. So, NAD plus, I am writing the reaction for link 2 pyruvic acid plus 2 CoA plus 2 NAD plus. Now, it gives rise to, it produces, what compounds are produced? 2 molecules of acetyl-CoA, each one having 2 carbons, right, plus, and at the same time 2 molecules of NADPH2 is produced and 2 molecules of carbon dioxide is released out. So, with the help of an enzyme, what is enzyme? Pyruvic dehydrogenase. Same link reaction what I have showed you here for explanation, and that is what we are writing in the form of a reaction. 2 molecules of pyruvic acid combines with 2 molecules. Okay. So, what is the thing we are writing here? 2 pyruvic acid joins with 2 CoA plus 2 NAD plus what is the enzyme? Pyruvic dehydrogenase multi-enzyme complex produced with 2 acetyl CoA having 2 carbons plus 2 NADPH2 along with the 2 molecules of carbon dioxide is released out. So, very important is this. So, here this step plays very important role because it brings acetyl CoA whatever is produced here. Now, this acetyl CoA enters into mitochondrial matrix. So, glycolysis happens in the cytoplasm, and here mitochondrial matrix acetyl CoA what is produced enters into mitochondrial matrix. From here it continues Krebs cycle. So, what is acetyl CoA here produced? Now, this continue the third step as Krebs cycle. Acetyl CoA what is produced end of link reaction. Now, it enters into and participates further with Krebs cycle. Now, we have to continue our discussion with what? We have to continue our discussion with Krebs cycle. Very important. So, I will just quickly revise it. The second step of aerobic respiration is called link reaction. Itself is also called oxidation 2 and decarboxylation number 1. Very important in remembering this 2 oxidation 2, and remember importantly decarboxylation number 1. Sometimes question occurs, where does decarboxylation number 1 occurs? That is in the conversion of pyruvic to acetyl CoA, that is in link reaction, right. As glycolysis occurs in the cytoplasm, acetyl CoA enters into mitochondrial matrix, that is why it is called link reaction, that is why this is also called as gateway step. Now, if you look at here, 2 and 1 are very important to remember. Now, let us see the reaction. 2 molecules of pyruvic acid, here end product of glycolysis, converts into 2 molecules of acetyl CoA. Out of 3 carbons, 1 carbon is removed in the form of carbon dioxide. So, 3 carbons will become 2 carbons, as here 2 molecules are there. So, there is a release of 2 molecules of carbon dioxide. Now, here there is a participation of 2 CoA, 2 CoA is added here, the 2 CoA is added here, added into the reaction. At the same time, 2 molecules of NAD plus get reduced to 2 NADPH2 with the help of magnesium and with the help of an enzyme. What is the enzyme which plays an important role here? Dehydrogenase. NAD plus converts into NADPH2 with the help of pyruvic dehydrogenase, right. Similarly, there is a release of 2 molecules of carbon dioxide. Which enzyme plays an important role here? Pyruvic decarboxylase. Pyruvic decarboxylase, and who transfers pyruvic to acetyl CoA? Third enzyme, trans-acetylase, right. So, here magnesium is also an important cofactor. 2 CoA is added, that is why the component what comes out is acetyl component, but that combines with CoA. So, we call acetyl CoA, that enters into mitochondrial matrix. But this step is being catalyzed by a multi-enzyme complex. What is this multi-enzyme complex? What is the name of that? Pyruvic dehydrogenase complex. Now, this multi-enzyme complex includes combination of 3 enzymes and 6 cofactors. What are 3 enzymes we can take? Pyruvic decarboxylase, first enzyme. Dihydrolipoyl trans-acetylase is the second enzyme. Dihydrolipoyl dehydrogenase, all 3 enzymes join together and they work efficiently with the help of 6 cofactors. What are the 6 cofactors? TPP, thiamine pyrophosphate, Second cofactor is lipoic acid. Third cofactor is CoA, that is coenzyme A. And fourth one is FAD. What is FAD? Flavin adenine dinucleotide. And NAD. NAD means what? Nicotinamide adenine dinucleotide. And the sixth cofactor is magnesium. 6 cofactors are needed for the functioning of 3 enzymes. That's why it's called multi-enzyme complex. What is that enzyme complex called? Pyruvic dehydrogenase complex. Right? Now altogether putting here, the total step of reaction includes, we write in single step reaction, that is 2 pyruvic acid plus 2 CoA plus 2 NAD plus on with the help of pyruvic dehydrogenase multi-enzyme complex produces 2 acetyl CoA plus 2 NADPH2 plus 2 carbon dioxide molecules are released out. At lastly, end of aerobic respiration, I will ask you the count of carbon dioxide and in which step they are released and where carboxylation, decarboxylation has taken place. I will ask you, you should be perfect because questions mostly comes with the balancing of the, which are the steps, where does it takes place and what is get released out. So in single step, this is a link reaction. Now what is produced here is acetyl CoA. Now it enters into where? Mitochondrial matrix. Now acetyl CoA, next it enters into the next pathway called as Krebs cycle. So now we have to continue our discussion with third step called as Krebs cycle. So before we discuss about Krebs cycle, let us discuss something else important. What is produced here in link reaction? NADPH2. NADPH2 produced where? In the mitochondrial matrix, right? NADPH2 produced in mitochondrial matrix. Now let us note down note points. What are note points? Note points are nothing but very important questions, question getting points, questions. Note points in the sense, let us take questions. Now if you take which questions will come? Glycolysis. Glycolysis, you remember there is a production of, so for conversion of where G3P2 1,3-biphosphoglyraldehyde, there is a production of NADPH2. NAD gets converted into 2 molecules of NADPH2. Did you remember? Right. Now what this is first step, glycolysis occurs where? In the cytoplasm. Very important. Now second step. What is the second step? Link reaction. In link reaction also, in conversion of what? Pyruvic acid gets converted into what? Acetyl CoA. Now what is produced here? NAD plus converts into 2-NADPH2. Now, so I will just make out very clear difference, listen carefully. Now this is a what story you know. This is a story we have studied and we have completed up to here. In glycolysis occurs in cytoplasm. Similarly, where link reaction occurs? Link reaction occurs in mitochondrial matrix. Mitochondrial matrix. See the difference. In glycolysis also there is a production of 2 molecules of NADPH2. In link reaction also there is a production of 2 molecules of NADPH2. But what is the difference? This NADPH2 what is produced here? It is produced in the cytoplasm. This NADPH2 what is produced here? It already entered in mitochondrial matrix, produced in mitochondrial matrix. What is the difference? Here each NADPH2 is equivalent to contributes for 2 molecules of ATP. That means energy. What is the energy you will get from here? What is the energy you will get? The energy from 2 NADPH2 equivalent to, that is 2 into 2 ATP. So you will get 4 ATP. Very, very important. That means 2 molecules of NADPH2 contributes and produces 4 molecules of ATP. Why? Because each NADPH2 produces 2 molecules of ATP. Why 2 molecules of ATP here? Because it's happening in the cytoplasm. Right? Similarly, coming to here, in this case, I am writing here each NADPH2, where it is produced in the mitochondrial matrix. Now it contributes for 3 molecules of ATP. Why 3 here? Why 3 here? Because it's produced in mitochondrial matrix. Very, very, very important difference. Here, out of 2 NADPH2 produced in glycolysis, each NADPH2 contributes or produces 2 ATP. Why 2 ATP? Because here NADPH2 produced in the cytoplasm, whereas the NADPH2 produced in the link reaction, each NADPH2 contribute for 3 ATP. That means energy, how much you will get here? 2 NADPH2 means here 2 NADP, it means 2 into 3 ATP. Altogether it contributes 6 molecules of ATP. So here 2 NADPH2 contributes for 6 molecules of ATP. Why 6 here? Because this reaction is taking place in mitochondrial matrix. Very, very, very important difference. Okay, this difference is very, very important. Remember, glycolysis, cytoplasm, because each NADPH2 contributes 2 ATP, the energy produced from 2 NADPH2 is 4 ATP because the reaction is taking place in the cytoplasm. Whereas NADPH2 produced in the link reaction, energy produced is 6 ATP because each NADPH2 contributes for 3 ATP because that is taking place in mitochondrial matrix. Very, very important difference, very, very important note points. Mostly you will get it in the form of a questions.